Good morning, children. So I hope you all have seen the yesterday's video, and where I've explained you about the nutrition. There we learned about autotrophic and heterotrophic nutrition, and we learned the definition of photosynthesis. And in that I told you that in the photosynthesis, the main raw material is carbon dioxide, and this carbon dioxide enters into the plant through the stomata, which are present on the leaves. They are also present on the stem also. Okay. See, stomata not only are present on the leaf, but few stomata are also seen on the stem part, delicate parts of the stem. Now, today we'll discuss about the cross-section area of a leaf. Before we study the importance why carbon dioxide, sunlight and chlorophyll are necessary for photosynthesis, before that we should know the structure of the cross-section of a leaf. Okay. Now here you can see I have kept a potted plant here, and in this you can see this is the main stem, and inside there are the roots, and through this there are the leaves present. And you have already studied about the plant tissues in 9th standard, where we learned about the complex types of tissues. And in complex tissues you learned about the xylem and phloem vessels, and these xylem and phloem vessels are continuing from the root system. Okay, so from the root system the xylem and phloem is been continuing, and it runs inside the stem also, and through the stem it will run into the leaves. So you have learned the main role of xylem and phloem. The xylem role was, that is the xylem role is to supply the water. It absorbs mainly the water, and all this water is absorbed through the soil, and that is how there is a pressure built up, and that is because of this pressure built up, the water will move from one cell to the other cell and reach to the tip of the plant. So there is a continuing channel, just as the water comes in our home through the pipe system. That is, from the rivers it reaches, it has been stored in the dams, and from the dams through the big pipeline system, that is how it reaches to our homes through small pipeline. So you can say in the same manner in a tree also, this xylem and phloem is like a pipe-like system which is supplying the water. Okay, and similarly the food which is prepared. Yesterday we learned in photosynthesis that photosynthesis process where the food is being prepared, and that food has to be supplied. See, that food which is prepared in the leaves is not only used by that leaf itself. It's not like that. We have seen that some amount of it has been stored in the form of starch, and that food is also been transported to the other parts of the plant through the another vessel called as the phloem. So phloem is taking that food which is prepared to all the parts of the plant system here, even to the roots. In xylem, the movement is always towards the upward, but in phloem the movement can take place in both upward as well as downward. Okay, so these things you have already learned in the 9th standard. Now let us have a close look towards this leaf. Now see at this leaf here. Now here you can see this leaf. Okay, here you can see this is a leaf here, and this is the petiole of the leaf, and through this petiole you can see there is a mid vein. Okay, this is a mid vein here. Okay, and these are the sub veins. You can see here these are the sub veins. Okay, and these mid vein and the sub veins, they are mostly compromises of the vascular bundle. Now what is vascular bundle? Vascular bundle you have learned in 9th standard. Vascular bundle is nothing but the xylem and made up of xylem and phloem vessels. Okay, so vascular bundle, what is vascular bundle? Vascular bundle is made up of xylem and phloem vessels. So you can see this mid vein. Okay, so this mid vein compromises of the xylem and phloem vessels, and on this leaf surface, this is the upper epidermis, you can say the upper layer of the leaf, and this is the lower layer of the leaf. Okay, and I told you yesterday that stomata are present on both upper as well as lower, but more stomata you will find mostly on the lower surface. You will see it on the lower surface rather than the upper surface. So how to see the cross section area of a leaf? See, actually now when the school will start, I'll show you the preparation of this slide, but at present only I'll give you the details of it. When you observe the cross section of a leaf, how does it look? So the preparation we'll do later, but now see here, if you have to take with the help of a blade, you can, now I'll take one more leaf, I'll show you. This is the Nerium leaf, the Nerium plant leaf, and in this Nerium plant leaf also you can see there is a mid vein and there are many side sub veins. Okay, so what you do is you have to take a cross section area in this manner. So this way you have to cut with the help of the blade, a transverse section. So in this manner, if when you take a transverse section in this manner, okay, you have to take a transverse section in this manner. You have to cut with the help of a blade in this manner, and very thin slice you have to cut that. And when you observe this part, okay, that means you have to cut in this manner, thin slice of it, a transverse section you have to take it in this manner. And when you cut in this manner, you will see, this is how you will see when you cut this leaf. Suppose this is a leaf, okay, now this is a leaf here, and you have cut the leaf in this manner, a transverse section, a very thin slice you have taken. This manner you have to take a thin slice of it. This is how you have to take a thin slice. Okay, this is a thin slice of that you have to cut in this transverse section. It's a TS of leaf, or you can also call it as a cross section of a leaf. And when you observe it on a microscope, when you illuminate it, you will see certain sections of it. Okay, when you just zoom it or when you observe it under the microscope. So how does this TS of a leaf look like? Let us see here the main parts of the leaf here, and then you will understand where the stomata are present more. You will find the stomata are mostly seen at the lower side of a leaf. As I told you, stomata will be present on the lower surface of a leaf. Okay, so if you see here, if I tear this is the lower surface, this part. So when I tear this, when I just now I just tear this leaf like this in this manner, and you can just see here, I'll just zoom it a little closer. So you can see now here this part of it, a thin part of the leaf. Okay, is it clear to you all? Yes. Now it is clear. Yes. So this part, this transparent part which you can see, we can take this transparent thin part of that, and when you observe it, this part, okay, this part, yes, see here, this part, this thin part, when you observe it under the microscope, you will see the lower epidermis consists of the stomatal pores. So before I describe the structure of stomatal pore, now let me explain you the cross section of a leaf. Now for a cross section of a leaf, what you do is, now today you will practice this diagram at home. Okay, so what you do is you draw 5 curves like this in this manner. This 1, 2, 3, 4 and 5. Okay, let me use the pencil. Sorry, I'm using the pen here. 1, 2, 3, 4 and 5, and then I'll make like this. I'll join this in this manner. So you make the cells in this manner. So these are the epidermal cells. This is the upper epidermis. These are the epidermal cells. Okay, and these epidermal cells is covered by a waxy coating. Now this is called a cuticle. You can also increase one more cell if you want. Okay, okay, so I've increased one more cell like this here. So this is upper epidermis. Now below I'll draw a lower, this side again below also I'll do. So you have to keep some distance in this manner and draw same. I'll do 3 curves this side. Okay, in this manner like this, and then I'm drawing this like this. Okay, got that? So, and leave little space here, and draw 2 more cells in the same line. and below also you show the cuticle. Okay, and now these are in the lower epidermis. Now you can show this is the stomatal. These are the guard cells. These are stomata. Okay, now these are the stomata, and here you can show. Let me use the sketch pen for you so you'll understand. Now here, so I'll draw the dots here with green color. That shows it has the chlorophyll pigments. See, only the stomata consist of the chloroplast, okay, whereas the epidermal cells does not have chloroplast. Make it clear, epidermal cells does not have chloroplast, okay, and only the stomatal pores, the guard cells, have the stomata. And here this is the nucleus. Okay, the cells will have the nucleus, so I have drawn with the pink color to show the nucleus. Okay, now, so as I told you, let me label this, the two parts which I have drawn. This is the upper epidermis, and this waxy coating is called as the cuticle. There's a waxy layer, you can say waxy layer or waxy cuticle. Okay, cuticle. Okay, similarly here also, this is the lower epidermis. Okay, and here also this is the cuticle, and these is the guard cells. These are the guard cells, or you can say the stomata. This is the stomata. Okay, this is the stomata. And now this part of the leaf which is present. Now when you're zooming, see this part, when you're observing it under a microscope, okay, you're observing it under a microscope. So this is how you will see under the microscope. That is how you're seeing a zoom part of this cells. So now between these two cells, there is number of cells which are sandwiched between these two layers. This is the upper layer of the leaf, and this is the lower layer of the leaf. Okay, and between these two layers there are many cells present, and these cells are called as the mesophyll cells, or they are called as a mesophyll tissues. Now the first layer of mesophyll tissue is perpendicular. Now you have to draw like this perpendicular cells. These cells are perpendicular to the upper epidermis. Okay, so you can see these are the cells. These are, you can say, oval shaped cells, long oval shaped cells which are perpendicular to the upper epidermis. Okay, so in this manner you have to draw a single layer of cells I have shown you. Okay, drawn here. So, and these cells also have a larger vacuoles in it. So these are the vacuoles. You have learnt in 9th standard about the plant cells. Okay, this is how I show the vacuoles in these cells. And now, okay, this is a perpendicular. This is one layer of cells, and this layer of cells is called as the palisade tissue. Okay, what is it called as? It is called as palisade tissue. It's also called as palisade parenchyma. You have learnt about parenchyma cells in 9th standard. So it's a one type of parenchyma cells. Okay, so it is called as palisade tissue, or palisade parenchyma also we can call it. So it is called palisade tissue or palisade parenchyma. And this palisade tissue consists of many chlorophyll pigments. You will find abundance of chlorophyll in these cells. So I am drawing with a green sketch pen over here dotted structures which represent the chlorophyll pigments. Okay, so this is how I have drawn the chlorophyll pigments here. Okay, so you can see here these are the chlorophyll pigments. Yes, okay. Now after this, below this palisade tissue, now there are loosely arranged small oval shaped cells, okay, in this manner. So they are not arranged in some pattern, they are arranged in some loose manner, okay. So you can see there are a lot of air spaces now. Here you can see there is a lot of air spaces. So this is how it is been arranged, loosely arranged cells. In this manner you will see these cells here. Okay, so this is how you see the cells here in this manner, and you can see there are air spaces between them, and these loosely arranged cells also has some vacuoles like this. But in them the chlorophyll pigment will be very, very less compared to the palisade tissue. So in these cells the chlorophyll pigments will be very less, few. So I will just show very few of it. Okay, compared to palisade tissue, these cells, these loosely arranged cells, are called as spongy cells or spongy tissue, or you can also call it as spongy parenchyma. Okay, so in spongy tissues very less chlorophyll cells are present. Okay, okay, so here you can see very less number of chlorophyll pigments are present, and this type. So what is this tissue called as? This tissue name is spongy tissue or spongy parenchyma, and both together make the mesophyll tissue. Both together is called as the mesophyll tissues. Okay, so mesophyll tissues are two types, palisade and spongy. So this is all together we see both of these present in the leaf. And these are, what are these green color dots which I have drawn here? These are the chlorophyll pigments. Okay, these are the chlorophyll pigments. And in the epidermal cells, both upper epidermis and lower epidermis, the chlorophyll pigments are present only in the stomata. Just remember it. Okay, epidermal cell lack chlorophyll pigments. That's very important, and it is covered by a waxy coating, a cuticle. Okay, and the chlorophyll pigments are present only in the mesophyll tissue, and more it is found in the palisade tissue, and it's very less in the spongy tissues. And these gaps which you see, what are these called as? The air spaces. So you see there is air spaces, okay, present between these cells. Okay, and that's why I told you that stomata are mostly found which surface? Lower surface, the lower epidermis compared to the upper epidermis. Okay, and when the sunlight falls on the leaf, when the sunlight falls on the leaf there, so this is a cross section of a leaf that we are seeing. So whenever the sunlight falls, all these chlorophyll pigments will move towards the upper side. Okay, they will move towards the upper side, and they get arranged here at the surface. For what? Why they are getting arranged? Yesterday we have studied about photosynthesis. What is important for photosynthesis? The sunlight. And that is why, and that is how the chlorophyll, what is it doing? It is trapping the sunlight, and it will prepare the food. And through this stomata, when the stomata is open, the carbon dioxide can enter. Okay, exchange of gases takes place. Whenever the stomata is open, exchange of gases will take place. So it can be any gas. Oxygen can enter, carbon dioxide can enter, it can come and go out, and even the water can also escape out. Okay, so for photosynthesis, which gas is important here? Carbon dioxide. So when the stomata open, carbon dioxide enter, and with the help of sunlight, okay, and the water which is present, it will prepare the food. So this was the cross section of leaf. So today you all draw this structure of the cross section of leaf and practice at home. And now we'll study, okay, tomorrow I'll teach you about the structure of the stomata. Now for today I think this is enough for you all. Okay, children. So tomorrow we'll learn about the structure of stomata, and for today you all just prepare with this structure of the leaf. And one more thing, I know that most of you are not having the textbooks, so it will be difficult for you. So as I told you, you can download the NCERT app. Okay, and from that NCERT app you can see the science textbook and the maths textbook. You can read from it there. Okay, so that will be easy for you and you can go through your lessons. And one more thing, I think you all are doing your homework, you all are revising your answers, whatever I am giving you, and I think you all are understanding what I am explaining you. Okay, if you don't understand, go through the video 2-3 times so you will understand it. Okay, thank you children, goodbye.